Many T cells respond to superantigens. Superantigens are a distinct class of antigens that stimulate a primary T cell response similar in magnitude to a response to allogenic MHC. Such responses were first observed in mixed lymphocyte reactions using lymphocytes from strains of mice which were MHC identical but otherwise genetically distinct. The antigens provoking this reaction were originally designated minor lymphocyte stimulating antigens, or MLS, and it seemed reasonable to suppose that they might be functionally similar to the MHC molecules themselves. We now know that this is not the case, however. The MLS antigens found in these mice strains are encoded by retroviruses, which have become stably integrated at various sites into the mouse chromosomes. They act as superantigens because they have a distinctive mode of binding to both MHC and T cell receptor molecules that enables them to stimulate very large numbers of T cells. Superantigens are produced by many different pathogens, including bacteria, mycoplasmas, and viruses, and the responses they provoke are helpful to the pathogen rather than the host. Superantigens are unlike other protein antigens in that they are recognized by T cells without being processed into peptides that are captured by MHC molecules. Indeed, fragmentation of a superantigen destroys its biological activity, which depends on binding as an intact protein to the outside surface of an MHC class II molecule, which has already bound peptide. In addition to binding MHC class II molecules, superantigens are able to bind the V-beta region of many T cell receptors. Bacterial superantigens bind mainly to the V-beta CDR2 loop and to a smaller extent to the V-beta CDR1 loop, an additional loop called the hypervariable 4 or HV4 loop. The HV4 loop is the predominant binding site for viral superantigens, at least for the MLS antigens encoded by the endogenous mouse mammary tumor viruses. Thus, the alpha chain V region and the CDR3 of the beta chain of the T cell receptor have little effect on superantigen recognition, which is determined largely by the germline encoded V sequences of the expressed beta chain. Each superantigen is specific for one or a few of the different V-beta gene segments, of which there are 20 to 50 in mice and humans. A superantigen can thus stimulate 2 to 20% of all T cells. And that's many T cells respond to superantigens. Have a great day.